Alrighty, so I wanted to cover just a quick lab final practicum review. So the lab final practicum, essentially what it's designed for is it's almost like a lab final basically, to cover some of the things that we would have covered in lab. Now, of course, lecture will have extended in the lab, and there's, lecture material, which is very, very similar to the lab material too, right? Um, so this is going to cover various chapters, various topics that we would have just simply covered throughout the whole course, basically. Okay. So starting off with digestion. So I know in week 8 we talk about the digestive system, and so we have to understand. Even in week 1, 2, we talk about some of the major macromolecules, right? These major biomolecules of the body, and so you have to understand what are the 4 major biomolecules and what are their building blocks, right? And so here on this slide, I give you an example. But before I go into that, I give you the review page too in your lab manual. Make sure you're paying attention to these review pages in your lab manual and be able to refer back to this when you're studying and preparing for your lab practicum. Now, when we're looking at the matching the end products of digestion with their types of food, so you're going to break down fats into fatty acids. You're going to break down carbohydrates into simple sugars. And you're going to break down proteins into amino acids, right? So you're going to be able to digest these different products and break them down into their monomer constituents. So we also want to be able to match the mechanism of absorption to the target effect or target effects of the following substances. So when we look at sodium, chloride, calcium, things like that, they're going to be absorbed in the body via ion active transport or via ion diffusion, right, down their gradient. And so fatty acids and glycerols, these are lipid molecules, so they'll be able to easily be absorbed into the body via diffusion, right, or through a cell membrane via diffusion. So we also have these enzymes too, such as secretin, right? So secretin will be able to stimulate the liver to secrete bile. Then we have amino acids, they'll be uptake via active transport. We have cholecytokinin. And so this is an enzyme that will go to the gallbladder and cause the gallbladder to actually contract to release its bile. And then, of course, monosaccharides, they're going to be absorbed via mostly active transport. And so when we want to match these digestion enzymes down here, match the following enzymes with their information, right? And so in the oral cavity, what are you going to have? You're going to have salivary amylase in the oral cavities. That's going to help to break down the carbohydrates. How are fats broken down via lipase activity? In the small intestines, that's where you're going to find this trypsin and this lipase helping to break down lipids and helping to break down proteins. Proteins are going to be broken down by trypsin. So trypsin is an enzyme that's able to break down proteins, right? Okay, it's like peptidases. These are other enzymes that are able to break down peptides, whether it's inside the peptide, right, like an endopeptidase or an exopeptidase, where it's going to cut at the ends of a peptide, for example. And these starches are going to be broken down by, again, amylase, right? So also understand cell division, right? How the cell will basically make a copy of itself, right? And so understand the different phases. And now these are just basic phases or basic explanations of what's happening in each phase, but you have to understand what's going on. So in interphase, that's where the cell is going to remain in interphase for most of its life, right? And so it's just doing its normal thing, its normal process, its normal function. And so also in interphase, we do note that DNA synthesis is occurring. Okay, so we have DNA synthesis occurring there. And then we have prophase, right? And so some books will say early prophase or prophase 1, prophase 2, right? Or early prophase and late prophase, I should say. If we're just simply looking at prophase, right? There's a couple of things that's happening in prophase, right? So notice this nucleus, right? You have this chromatin right there in interphase, but notice how it's condensed right there. So in prophase, we have the condensation of these chromosomes, or these chromosomes are being condensed. We also have that nuclear envelope going away, so it's disappearing. And we also have these mitotic spindles forming, right? That's coming from these centrioles. And so they kind of push apart, push on each other as they're going to migrate toward the opposite poles of the cell. So that's so you can see these microtubule organizing centers, these centrioles, at the opposite poles of the cell. In metaphase, or prometaphase and then metaphase, essentially what's happening is these microtubules, they're attaching to the kinetochore protein of the chromosome, and so they're aligning these chromosomes up on the metaphase plate. So that's what we have in metaphase. These chromosomes are literally lined up in the equator on the center of the cell. And then in anaphase, notice how they look like V's. They're shaped like a V, right? Because these chromatids or these sister chromatids, they're literally being pulled apart to opposite poles of the cell. And then in telophase, so telophase is almost essentially like I guess we can say the opposite of prophase, right? Because now, and I wish I showed this in an image, kind of can see it like this, because now these chromosomes, they are being uncondensed now. So they're unraveling back into this chromatin formation. Also, we have in telophase, the nuclear envelope is starting to reform, and we have this cleavage furrow. This cleavage furrow is starting to burrow in, and it's basically going to separate this cell, making it two identical daughter cells, and that's where we have cytokinesis. So if we look here, honestly, in this phase right here, right, this is mitosis, right? This is basically what we call, or we can identify this as nuclear division, because we're dividing the nucleus. What's in the nucleus, the chromosomes. So we're literally dividing them, we're pulling them apart, as you can see right here. So that's why we call mitosis nuclear division, whereas cytokinesis, because that cleavage furrow was being developed in telophase, cytokinesis, this is what we call cytoplasmic division, because we're simply dividing the cytoplasm, right? So understand what's happening in these phases and understand under a microscope what they kind of look like, okay? So understand tonicity as well, right? So we talked about tonicity. So water tends to move from a hypotonic solution to a hypertonic solution. What does that mean? Water simply wants to move to where there's a greater amount of stuff at, okay? So tonicity, the movement of water, really depends on these non-penetrating solutes, okay? So water is going to follow wherever there's a lot of solutes. So if these solutes can penetrate through the membrane, they're cross through, and then water would just simply follow them as well. So understand that if we place this red blood cell in an isotonic solution, and so again, tonicity refers to the change in volume of a cell. So what will that cell look like in this specific solution? So if I put the cell in an isotonic solution, that cell remains the same shape. There is no change, right? So no change to the cell shape or size, because equal amounts of water go in and equal amounts of water go out. If I put that cell in a hypertonic solution, meaning the solution has more than the amount of stuff in the cell, water wants to move with their stuff, so water is going to move out of the cell. As a result, that cell is going to shrivel up, crenate, and die. If I put that cell in a hypotonic, it's going to look like a hippo, but if I put that cell in a hypotonic solution, the solution has less than the amount of stuff in the cell, and if the cell has more stuff, water is going to want to rush inside the cell, making it a hippo, or a large cell is going to swell up and lyse and burst. Understand the different functions of epithelial tissue too, right? So we have epithelial tissue for protection, such as your skin, your stratified squamous epithelium, right? Then we also have epithelial tissue for like diffusion. So we have like simple squamous epithelium, right? So one layer of these cells, and that's going to be your alveolar type 1 cells, right, in your lungs. So we also have epithelial cells that act for secretion, right? These are going to be those simple columnar epithelium, right, such as those glandular cells. We also have epithelium for filtration, right? And this is going to be those simple cuboidal epithelium, such as we're going to find them in the kidneys. We'll also find them in the digestive tract too, for like absorption and filtration, okay? So you definitely want to remember these different type of vision definitions, right? And so sometimes we'll cover it in class, sometimes we might not have the time to, but remember these different types of refractive errors that we'll see too. So we have normal vision, right? Normal vision is just emmetropia, right? Now, when we have like an astigmatism, right? And so you can see this astigmatism right here, and you can see that this is where we have blurred vision due to the unequal curvature of your lens or your cornea, okay? We have an accommodation. Accommodation just simply means your ability to focus on close-up objects. We have hyperopia, right? That we can see right here, hyperopia. That's going to be far-sightedness. And so if you're far-sighted, you have the inability to focus well on close-up objects, whereas nearsighted, which is like myopia, this is going to be nearsightedness, right? So it can happen in both eyes or one eye. So for example, me, I have left eye myopia, so I'm nearsighted in my left eye, but I guess my right eye is good. Also understand the definitions of refraction and convergence, right? Especially refraction. So refraction is basically light bending. And so whenever light goes through a certain medium, such as air or water, it actually bends, okay? And then of course, convergence is just that medial movement of your eyes during focusing on close-up objects. Also understand the different steps of a reflex arc and know these steps in order, okay? Don't get mixed up with these, okay? Also refer to these pages in your lab manual. It's going to start off with a stimulus, right? That stimulus is going to get picked up by a receptor. That receptor is going to send that information through a sensory neuron to the integrating center. From the integrating center, you're going to send out a motor neuron to the effector tissue to have some effect. So remember these steps in order. Sorry, let me go back to the Babinski sign. So understanding, we talked about the Babinski sign when we did like the reflex labs. And so understand that the Babinski sign is going to be an abnormal response to the plantar reflex at the bottom of the foot. And that's going to display unmyelination of neurons, okay? Now, this Babinski sign is going to be considered normal, a normal reflex in newborns due to their undeveloped myelin sheaths on their nerves. However, it's abnormal in adults, right? And in children over 2 years old, because they should have, the myelin sheaths formed on their neurons. But again, if we have this Babinski sign that's fanning out of the toes when you stroke the sole of the foot, it will display underlying nervous system or brain conditions, okay? Now, spirometry. Understand what spirometry is. We did that spirometry test, right, using the spirometer. So spirometry is basically a pulmonary function test to test the function of your lungs, the health of your lungs, essentially, right? So be able to understand what these values are, especially tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume, total lung capacity, vital capacity, right? Those are the ones we really talked about. And so you got to remember what they are and how to calculate them. So blood typing as well. So understand blood typing. Understand the importance really of blood typing too, right? You can safely receive blood transfusion or a transplant. You can find out a parent, right? Who's a parent or not, right? So blood typing has a couple of different uses, right? So understand just the blood type, right? And how we see this, how this manifests. So when we're looking at the antigens are these proteins on the surface of a red blood cell, and antibodies, they're going to be found in the plasma, okay? So if you're, for example, if you're blood type A, you're going to have A antigens on the surface of your red blood cell, but you're going to have anti-B antibodies in the plasma, right? Whereas if you're blood type O, you have no antigens on your red blood cell, right? But you do have both antibodies in your plasma. And if you're AB, you have both A and B antigens on your blood cell, but no antibodies in your plasma, of course, because these antibodies, when they target that antigen, they're going to clump up and they can cause hemolysis, okay? So understand the different parts of a EKG too. So we talked about this a couple of times, right? This electrocardiogram, it just shows the summed electrical activity of your heart cells. So it's not the same as an actual potential though, of course, but it does show the summed electrical activity of your heart. Now, there's going to be 3 waves, and so understand these waves. Again, P wave is where the atria will depolarize. This is where the atria will contract, right? The QRS complex is when the ventricles will contract, when the ventricles depolarize. This is also where the atria will repolarize, when they will relax as well, okay? And then we have the T wave. The T wave is when the ventricles will relax, when they repolarize, okay? So you got to remember these different waves of the ECG. When we look at blood pressure, right, and and heart sounds. So understanding, blood pressure, if it's 120/80, right, we have the systolic over the diastolic number, okay? When you're trying to auscultate or listen to a person's heart with that stethoscope, right? So typically when you're trying to measure blood pressure, you'll put that sphygmomanometer around the arm, it'll squeeze the arm, it'll give you pressure and readings of millimeters of mercury, right? And then you try to listen using the stethoscope. Now, when you're listening to the heart, right, there are these heart sounds that you got to remember, okay? And they really go along with like the valves of when blood enters the heart. So the first heart sound you're going to hear is the lub, right? And then the second heart sound is going to be the dub. So it's like lub dub, lub dub, lub dub, lub dub, right? And so the lub, the first sound, is coming from the closure of the AV valves, whereas the dub is coming from the closure of the semilunar valves. Of course, remember, when blood enters the heart, what does it enter through first, right? What valve, I should say, is going to enter through first out of these two? It's going to be an AV valve. So you can kind of remember that. Then it's going to go through a semilunar valve. So AV valve comes first, the semilunar valves come second. Now, blood pressure, we give like the classical reading, a good blood pressure is 120 over 80 millimeters of mercury. If you have elevated blood pressure, right, that's when your systolic number is going to be a little bit higher, 120 to 129 or 130, right? Diastolic number is going to be below 80. And understand these different stages of hypertension too. Stage 1, 130 to 139, and then stage 2, 140 or higher. Now, if this person is in a hypertensive crisis, right, this means they will require immediate medical attention. This is when your systolic number is higher than 180 and your diastolic number is higher than 120, right? So understand that about a hypertensive crisis. Okay. Also understand some of the factors that will influence mean arterial blood pressure that we talked about before. Your blood volume. So how much fluid do you have in your blood, in your body? If you have a little bit amount, your blood pressure is going to be low. If you have a high amount, your blood pressure is going to be high, right? Also, other factors that will determine blood pressure will be the effectiveness as your pump of your heart as a pump, right? That's going to give it its stroke volume, how much fluid is being pumped out each time. Your heart rate, how many times is it going to pump per minute? Also, the resistance, right? And the diameter, I should say, of your arterioles, the diameter of your veins, right? These are other factors that will affect your blood pressure. And also understand things that can increase your blood pressure and lower your blood pressure, right? So increase sodium, things like that. But we can decrease our blood pressure with, relaxation, no stress, exercise, things like that. Also understand the RAS system, the renin-angiotensin-aldosterone system. This system is stimulated when we have a drop in blood pressure. When we have a drop in blood pressure, we're going to release renin. And renin's job really is to raise blood pressure. How? So renin will act on the already circulating angiotensinogen that's coming from the liver. Understand where it's coming from. So renin will convert angiotensinogen into angiotensin 1. Angiotensin 1 travels around in the blood, goes across the lungs. The lungs have this ACE enzyme, angiotensin converting enzyme, that will convert angiotensin 1 to angiotensin 2. Angiotensin 2 will then go to the adrenal cortex so you can release aldosterone. Aldosterone goes to the kidneys, tell the kidneys to reabsorb sodium, right? So we can reabsorb water. Angiotensin 2 is going to go to the hypothalamus to tell you to release ADH. ADH goes to the kidneys, tell the kidneys to conserve water. Angiotensin 2 goes to your blood vessels, constrict your blood vessels, increase your blood pressure. All of these results is trying to do the opposite of that stimulus. That initial stimulus was a drop in blood pressure, resulting in what you want to do homeostatically is raise blood pressure. Urinalysis. Understand what we use a urinalysis for, right? To study the health of urine, study the health of your kidneys, I should say. And so the color should be yellow, pale yellowish, clear. Clarity, it should be clear, right? It shouldn't be cloudy that much, right? pH around 4.5 to 8. Specific gravity is basically how much stuff is in your urine, right? So if you're, just urinating a lot, right, over and over and over, you're not going to have that much stuff in your urine. It's not going to be concentrated. But if you're holding your urine, if you're not urinating enough, then it's going to be more concentrated when you finally urinate, right? So that's what specific gravity is, just the concentration of stuff in your urine. Shouldn't be no glucose in there, shouldn't be no ketones, shouldn't be no nitrites, shouldn't be no leukocytes in there as well, right? Also, ending up with endocrine. So understand endocrine system, right? And I won't go through everything, right? But understand the hypothalamic, pituitary, peripheral organ axis for all of these, right? And I'll just simply give you an example. We have thyrotropin releasing hormone that gets released, goes down that hypothalamic pituitary portal system, that hypophyseal portal system, right? Also understand the different portal systems too. So I'm gonna put a star right there. Say understand the different portal systems too. So TRH, thyrotropin releasing hormone, goes down the hypophyseal portal system to target the anterior pituitary to tell it to release TSH. TSH then goes to the peripheral organ, the thyroid gland, to tell the thyroid gland to produce T3, T4. So remember all of those other ones as well, and understand the different functions of them too. Okay? Understand what these organs, what hormones they're going to produce. Okay? Got to remember those. And I end with some thyroid and adrenal cortex disorder. So we talked about this before. If you have a case of hyperthyroidism, right, that's where you're going to have too much, for example, too much iodine, and you're making too much T3, T4. So hyperthyroidism is where you have the lab values, is where you have elevated T3, T4, but reduced TSH, though, right? That would be in the form of Graves' disease, right? Hyperthyroidism, okay? Your thyroid gland is just stimulated too much. And we have hypothyroidism, lab values. So understand these lab values, right? Hypothyroidism is where we have low T3, T4, right? But elevated TSH, right? When we look at the adrenal cortex disorders, we have Cushing's disease. Cushing's disease is when we have too much of these steroid hormones, too much of cortisol, okay? Whereas Addison's disease is where you have too little, right? So not enough. So understand the difference between the two, right? Happy studies.